Well, hello, everybody. It's really a pleasure to get to speak with you today. And thank you so much, Matt, for the kind introduction. Today, I'm going to cover a bit of work that we do in my group regarding IPM. And this is part of a broader research program in my lab that deals with developing and advancing sustainable food systems. And biosolarization, the technology I'll focus on today, is really at the intersection of many different sustainability concepts in food systems, from displacing the use of toxic conventional fumigants to creating circular economies in food where we use the residues from food production to promote production of additional food, to developing long-term soil health that will maintain the ability to productively produce crops long into the future. And as you heard, the focus of today is to look at the utilization of byproducts and residues from almond processing to advance IPM with a focus on almond hulls and shells. So if you've eaten almonds, the fraction that you're eating is really the kernel, which is part of a larger almond fruit. So in the course of processing almonds, there are several layers of that fruit, specifically the pericarp, that are removed and used for other non-food applications. These are the almond hulls and shells. So the almond hulls are really the exo and mesocarp fractions. They're a very leathery material, rich in fiber, some sugar and protein as well. And then the endocarp is the woody layer that surrounds the edible kernel. And the way that these residues are used currently is that most of the hulls are routed into use in livestock feed, and the shells are used for animal bedding, primarily in the California dairy industry. Now, there have been some fluctuations in the scale of the dairy industry with respect to the almond industry, and that's driven research to find new utilization opportunities for these materials. And that's where biosolarization comes in. And biosolarization is an IPM technology that represents an alternative to conventional soil fumigation. And the way that it works, and I'll dig deeper into the mechanisms in just a moment, are that it utilizes passive solar heating and soil fermentation processes to create multiple stresses on soil pests and pathogens. And so you do get pest control by virtue of those actions. And then you get some secondary benefits that I'll cover as well with regards to enriching organic matter in the soil and the litany of soil health benefits that go along with that. So I think it's worth reviewing how we conduct biosolarization in the field. So if we consider a plot of agricultural soil, what we would do is broadcast some organic amendment. In this presentation, I'll be focusing on the almond residues, but we have successfully deployed biosolarization using skins and seeds left over from California tomato processing, skins and seeds from winemaking, spent grain from brewing. There really are a menu of different organic residues from the wide variety of specialty crops grown in California that could potentially be used in this way. But after we broadcast the material on the soil, we then incorporate it through tillage or disking, smooth the soil, and then from there, we lay drip line or drip tape and then mulch with clear plastic film. And this is actually the same virtually impermeable film that you would use if you were doing fumigation, but here we're repurposing it for biosolarization. As we would do in fumigation, the edges are buried during tarp application to create a very close seal and minimal headspace between the tarp and the soil. But if we consider a cross-section of this setup, really looking at the interface between the soil, the tarp, and the atmosphere, we can start to deconstruct what's happening during the process and how the different pesticidal mechanisms work. So again, looking at these three layers, one thing that happens is that we have passive solar heating. So this is a clear, transparent tarp, so visible light is able to penetrate it. That's absorbed by the soil, but the soil is also radiating out light in the form of longer wavelength infrared radiation. The optical properties of the tarp are such that while it's transparent to visible light, it's actually reflective to the longer wavelength IR radiation. So that gets reflected back to the soil. So what you've done here is induce a greenhouse heating effect at the level of the soil, basically the same mechanism through which your car gets very hot if you park it outside on a sunny day. And so this solar heating method, also called solarization, can lead to appreciable increases in soil temperature, particularly at the topsoil layer, uh, exceeding even 65 degrees centigrade. So that type of heating alone can often be very effective at controlling pests in the topsoil. But we have some other things going on here by virtue of having amended the soil with organic matter and then irrigating it through that drip line to create a saturated environment. So the combination of the saturation and the tarp on the surface excludes oxygen from the soil, which means that microbes that are endogenous to the soil or are introduced on the amendments themselves start to hydrolyze and metabolize the organic matter in the amendments. And because it's a low oxygen environment, the form of that metabolism is anaerobic fermentation. So we have this accumulation of fermentation products, largely organic acids, things like acetic acid, propionic acid, butyric acid. Now, these are all known biopesticides. They are very effective at inhibiting a broad spectrum of soil pests, from nematodes to fungal and bacterial pathogens and even weed propagules. So you have that broad spectrum activity that you would have with a conventional fumigant. The key difference here is that these organic acids are much, much more innocuous for people compared to conventional fumigants. You know, if you think of the toxicity of something like acetic acid, it's pretty low. We deliberately consume that in vinegar. Propionic acid is something that is made by bacteria on our skin. And if you've ever kind of smelled not too great after going to the gym, what you're smelling largely is that propionic acid. And then butyric acid is a constituent of our stomach acid. So our bodies are producing it continuously. So when you compare that level of safety compared to a conventional fumigant, like chloropicrin, which is one of the most widely applied soil fumigants in California. Chloropicrin, before it was used in agriculture, was used in chemical warfare to clear trenches in World War I. Similarly, 1,3-dichloropropene, another very popular fumigant, raises serious concerns about atmospheric exposure and accumulation, and its both acute and chronic toxicity. So there's just a step change in risk by going with these organic acid biopesticides compared to conventional soil fumigants. So this is something we wanted to examine in the field. And so in 2017, we conducted a biosolarization field trial in collaboration with a commercial almond grower. This was the Nicholas Nut Company based out of Chico. They had a 50-acre plot that had previously housed a walnut orchard. They had tore that out and were in the pre-plant phase to establish an almond orchard on that site. Because there had been 50 years of walnut cropping, there was quite a background infestation of phytoparasitic nematodes, primarily ring and root lesion nematodes. And so we had a good basis here to examine the deployment of biosolarization to control those pests during the pre-plant period. So what you see here is a 10-acre section of that 50-acre plot that we used for our field trial. Within that 10-acre section, there were a number of different biosolarization treatments examined. So specifically, we had 2 biosolarization treatments that differed based on the amendments that we used. So one used almond hull and shell from the non-Perel variety of almonds. This is the most popular variety of almonds because it has a very large, attractive kernel, and the mass fraction of the kernel with respect to the overall almond fruit is quite high. It has a very thin shell, and so you get more return on investment, essentially, in terms of the amount of kernel you get per tree. We compared the utilization of the non-Perel hull and shells to biosolarization using hull and shell from other varieties. And if you are familiar with almond production, almonds cannot self-pollinate, so any almond orchard is always going to contain multiple varieties of almonds. So in addition to the non-Perel whole and shell, we used a mixture of pollinator variety whole and shell that would represent a combination of whole and shell from Aldrich, Monterey, Bennett Hickman varieties, all aggregated. And so different varieties have different physical and chemical properties to their whole and shell, which could have ramifications on biosolarization performance. So we compared biosolarization to two controls. One was a solarized control where we set up the plots with the drip line, with the tarp, but we didn't apply any amendments. So it was separating out the effect of the solar heating from the combined effect of the solar heating and soil fermentation stresses. And then we compared that to a true negative control, which was just untreated soil. So those are the 4 soil treatments. After those soil treatments were completed, then the trees were established using the pattern shown up at the top. So alternating non-Perel rows with different pollinator variety rows. In this case, it was alternating the non-Perel trees with either Monterey or Bennett Hickman variety. And we tracked multiple measures of soil chemical responses as well as subsequent tree responses after the orchard was established. So let's look at some of the short-term effects of biosolarization. I mentioned that organic acids produced through anaerobic fermentation are one known mechanism of pest inactivation. So one thing we did was to pull 12-inch soil cores from the treated plots throughout the biosolarization period, which lasted for about 6 weeks. And we looked at the total organic acid concentration within the upper 6 inches and then the subsequent 6 inches below that. And so what we can see here is that there were significant differences in the amount of organic acids that accumulated for each of the 2 biosolarization treatments, namely the soils that were amended with the non-Perel hull and shell yielded more organic acids in the soil compared to those that were amended with the mixed pollinator varieties. Another observation was that there was greater organic acid accumulation in the lower layer of soil, that's the 6 to 12 inches depth, compared to, actually I had that backwards, we had higher organic amendment accumulation in the upper 6 inches of soil compared to the subsequent 6 inches deeper down. And that's really probably due to the distribution of the amendment. We tried to have uniform incorporation of the material down to about 8 or 9 inches, but by virtue of the disking, it was probably still largely concentrated near the upper layers, and we see that reflected in the acid data here. So concurrent with the measurement of organic acids, we also looked at levels of viable root lesion nematode. So on day -1, which is the day preceding the start of biosolarization, you can see the background infestation, fairly uniform across all treatments. And then we took measurements at 9 and 41 days after the onset of biosolarization. And you can see by day 9, any of those solar heated treatments showed dramatic reduction in the root lesion nematode levels, and that persisted through day 41. If we zoom in on some of the data here, we do see some nuances insofar as at day 9, while any solar heated treatment showed very large reduction in nematode levels, if you look closely, the biosolarized treatments were essentially 0 in terms of viable nematode levels, where the solarized treatment still had a small but significantly non-zero infestation level. So it speaks to the fact that solar heating can do a lot to promote pest control, but there are some accelerated kinetics of phytoparasitic nematode inactivation by virtue of coupling those fermentative stresses on top of the solar heating. Certainly, if you're a grower, you'd like to see 0 root lesion nematode compared to a small but non-zero value. So that was the short-term data for nematode control. We were curious to see what the legacy effect of that would be. So we've been tracking nematode levels in the years since that 2017 biosolarization trial. Here are some data from 30 months after completion of the process. showing that in our solarized and biosolarized plots, we still see significant reduction in root lesion nematode levels compared to the untreated controls where the infestation really persisted. So I'd like to move on from the pest control data to talk about some of the secondary benefits of biosolarization, specifically benefits that come from improved soil health by virtue of having that organic matter incorporated into the soil as part of the process. So we tracked soil nitrogen. This is total nitrogen for several years after the biosolarization treatment. What we saw was that for the biosolarized plots, these were the amended plots, we saw a significant increase that was maintained for over 2 years in total nitrogen. You can see those data here. And then as we get into years 3 and 4 post biosolarization, we start to see those significant differences disappear. And that period will align with some interesting effects in tree growth, which I'll get to later on in the presentation. So there's a question of, what form of nitrogen is this? This is total. So looking at organic versus mineral nitrogen, we did separately track ammonium nitrogen and nitrate nitrogen. Really, the primary effect on the ammonium levels was during the biosolarization process. You can see that to the far left here, where any of the solar heated treatments showed significantly increased ammonium levels within the soil. This makes sense because at higher temperatures as we had here, and this was, approaching 140 degrees Fahrenheit near the surface, at those higher temperatures, ammonification would be favored over nitrification. And so we see this elevated ammonium levels, but it's only during that treatment period where we had the increased soil temperature, and we don't see any significant differences after the process is finished and the tarps are removed. Likewise, for nitrate levels, we see significantly lower nitrate levels during the biosolarization process. Again, nitrification would not be favored at those temperatures, and the anaerobic environment within the soil may actually promote a bit of denitrification, and so that could be another effect underlying these data. But again, once the process is finished, we don't see any significant differences in nitrate levels across the treatments. So when we go back and look at the total nitrogen, these significant persistent differences are likely due to significantly elevated organic nitrogen that is slowly mineralizing over time. So looking at another plant macronutrient, potassium, it's known that almond hulls are very rich in potassium, and we see that reflected in the soil data for the biosolarized plots containing these hulls and shells, insofar as there's significantly elevated potassium for over 2 years post-treatment. Again, once we get into years 3 and 4, those differences erode and we see more of a convergence. And again, I'll speak to how differences in tree growth may help to explain why we see the eradication of the significant difference. And then capping off the soil chemical characterization by looking at total carbon in the soil. As you might imagine, the soils contained organic amendments showed matter within the soil. And while this soil carbon itself is not a plant nutrient, it does affect different aspects of soil health that could promote plant health. For instance, the water holding capacity of the soil is dramatically increased by this organic matter. There could be beneficial effects to cation exchange capacity. The humification of the organic matter can promote beneficial microbial diversity and soil aggregation that helps to promote porosity and resist erosion. So there is a bit of a carbon sequestration effect for at least 2 years after treatment. But again, once we get into the latter years, 3 and 4 years post biosolarization, we stop picking up as dramatic differences in soil carbon levels between the different treatments. So moving on from the soil properties to the properties of the trees, one interesting effect that we saw was in the first year of growth, we didn't see any significant differences largely between trees that were grown in the biosolarized plots versus the solarized control versus the untreated control. And in fact, in some cases, we saw significantly slower growth for the trees that were in the biosolarized plots. You can see the growth rates over time for each of the 3 varieties in the orchard in response to growth within the different soil treatments. And so what this indicates is that there's likely some residual phytotoxicity within the soil after this process concludes. Now, we had been tracking organic acid levels, and organic acid levels, while speaking to biopesticidal activity, those same organic acids can be phytotoxins. Those organic acids alone would not explain these data because we had shown eradication of those organic acids pretty quickly after the tarps were removed. After you introduce oxygen back into the soil, those organic acids, which were produced through anaerobic activity, then become substrates for aerobic respiration, in addition to the fact that these are largely volatile fatty acids. So once the tarp is removed, they can also volatilize and escape the soil. So we did not have any detectable levels of organic acids in the soil that would explain either the lack of difference in tree growth or in some cases the inhibitory effect on tree growth. Now, probably this relates to some other chemical factor within the hull and shell. And it is known that almond hulls are very rich in various phenolic compounds like flavonoids, and some of these are known phytotoxins. So it could be that there's persistence of these compounds within the soil that could affect the short-term growth of different crops. But once we got past year 1, what we see in year 2 is the emergence of faster growth in some of the tree varieties grown within the biosolarized plots. So there are 2 things that could be happening here. One is that there's just been sufficient degradation of whatever phytotoxic compound was in the hull and shell, and so you no longer see that inhibitory effect. Or perhaps the trees simply adapted and acclimated to the soil conditions and then were able to exhibit increased growth. But we do see this very marked inversion of the growth rate trends, where once you get into year 2, trees that are within the biosolarized plots do start to show accelerated growth compared to the controls. And if we look at our most recent data from May of this year, which is over 4 years after the completion of biosolarization, we do see the effect of that accelerated growth in some of the biosolarized plots. Now, there's an interaction between the biosolarization conditions and the variety of almond that's grown within the treated soil. But the data that we have so far have shown significantly increased trunk diameters for non-Perel trees that are grown in soils that have been biosolarized with non-Perel hull and shell. So too do we see significantly increased trunk diameter for Monterey variety trees grown in soils that have been biosolarized with non-Perel hull and shell. And then for the Bennett-Hickman variety, we do not observe any difference in trunk diameter for any of our soil treatments. So again, showing that there's opportunity to tune the biosolarization conditions to different almond varieties to maximize the potential for a positive effect on the crop. So while the data do show that you can simply wait it out and get past that 1 year potentially inhibitory period post biosolarization, we would really like to eliminate or at least dramatically reduce that period so that we can get to observable benefits to plant growth as quickly as possible. So we've done some additional experimentation to look at process variables that growers can control to see if we can really reduce the amount of remediation time, which would be the time between the conclusion of biosolarization and the time at which you start to see a positive effect on plant growth. So one process variable is simply the amount of amendment that you apply to the soil during biosolarization. So here's a study where we looked at 2 levels of non-Perel hull and shell applied to the soil in simulated biosolarization, 2.5% by weight and then 0.6% by weight. And then we tracked the remediation of the soil over 4 weeks. And the metric for gauging phytotoxicity here was a growth index measurement. Basically, you take samples of your soil, you do an aqueous extraction, and then you apply that extract to radish seeds in a petri dish, and then you measure the germination and radical growth of those seedlings to compare against a control with just water and calculate this growth index. So if you have a growth index of 100, you've matched the growth of the positive control. Anything below that would suggest some level of inhibition to the plant. So the data show that if you amend the soil with less material, you can dramatically reduce the amount of remediation time, dramatically reduce that inhibitory period. So there's a significant difference, for instance, between soils with a 2.5% application rate and those with a 0.6% application rate. And so what this suggests is that there is an optimum in terms of the level of material applied to make sure that you're getting that biopesticidal effect, but not going so far as to overload the soil with potential phytotoxic compounds and prolong that adaptation period before the trees can show accelerated growth. Another process variable that growers could control is the particle size of the amendment. So in that field trial, we used a very coarse particle size. While we did run the material through a tub grinder, the average particle size was 2 centimeters, so almost unchanged from how it came out of the processing facility. And so what that did likely is prolong the degradation time for the material in the soil after biosolarization. So we did a study using simulated biosolarization in the lab to look at the effect of particle size on the remediation period, or how long it would take for the soil after removal of the tarp to show a growth index value that matched a water control. So you can see some interesting effects here insofar as there's differing effects of particle size on the hull and shell that come from different varieties of almond. Specifically, the non-Perel hull and shell do show a very dramatic and significant benefit to using a finer particle size to promote more rapid degradation of any phytotoxins and thus dramatically reduce the remediation period after treatment. So based on these data, you would want to use something that's almost like a fine flour. So investing that additional energy to grind the material down to that texture may yield a net benefit by reducing the potential inhibitory period after biosolarization. Conversely, we don't see really any detectable effect of particle size on the pollinator residues, so you could get away potentially with a more coarse particle there. So presented some data showing the effects of biosolarization, which were ultimately positive on tree growth as indicated by trunk diameter. We've also been tracking tree health using multi-spectral imaging. So this is an aerial imaging technique that measures the absorbance of the canopy at various wavelengths and compares different ratios for these absorbance values that correlate to different aspects of tree health, stress, and activity. So this is something that we've done annually in the summer at the site. And so different trends have emerged over time from the multispectral imaging. If we look at May 2020, which was about 2.5 years after the trees were planted, that was the first time we started to see some significant differences between the tree canopies based on the soil treatments. Specifically for the non-Perel trees that were biosolarized in the soils that were treated with the non-Perel hull and shell. And these were trees that we had observed a significant increase in trunk diameter for. We also see significant greater absorbance values that correlate to how green the canopy is. So this is indicating that both the trees in this treatment have more vegetation, but within the vegetation, they also have a greater concentration of chlorophyll. Now, these differences become more expansive if we look out an additional year to June 2021. Now we start to see additional differences between other varieties grown in biosolarized soils with respect to the controls. For the Bennett-Hickman trees established in the biosolarized plots containing the pollinator variety hull and shell, we see significant increases in absorbance ratios that speak to the amount of vegetation and chlorophyll and photosynthesis occurring, suggesting that the trees in the biosolarized plots were more photosynthetically active. Likewise, for Bennett-Hickman grown in the biosolarized plots containing the nonpareil hull, we also see elevated green values that suggest increased concentrations of chlorophyll. And then for the Monterey variety of tree grown in the biosolarized plots containing nonpareil hull and shell, we also see these absorbance metrics that speak to elevated chlorophyll. So the thought is that we have multiple metrics that align to suggest that the trees are growing more vigorously and that they may be more metabolically active. And the hope is that these are early signals that ultimately the trees will be more productive and show elevated yield for those grown in the biosolarized soils. And that's where we're at now. So this summer, we're going to get our first yield data from the orchard. And we have high hopes for those data based on these early signals, but that's really the ultimate metric for us to show that biosolarization, in addition to the long-term phytoparasitic nematode control, in addition to the enrichment of plant nutrients, and in addition to benefits to the tree growth, that ultimately that translates to significant elevation in yield within these biosolarized soils. So, I've framed biosolarization as a sustainable alternative to fumigation because it displaces the use of toxicants. It puts food waste really to use for us in agriculture, and it improves soil health in a number of ways by virtue of promoting organic matter enrichment and persistent elevation to plant nutrients in the soil. But can we make a quantitative claim on the sustainability of biosolarization compared to fumigation? So we've done a life cycle assessment, and in this study, we were looking at the scenario where we took the tomato pomace from industrial tomato pomace processing, which are the skins and seeds from the tomato fruit. What if we diverted all of that away from its current use in animal feed and instead put it to use in biosolarization? Now, while this is an LCA for biosolarization with tomato residues, tomatoes have a number of commonalities with almonds insofar as the production is heavily concentrated in the Central Valley. The current use of the material is in animal feed, and they are compatible with biosolarization. Excuse me. So without getting into all the details of the LCA, we did compare use of the tomato residues in biosolarization, which would divert them from animal feed and require substitute feed components, to the business as usual case, where the pumice would be used for animal feed and soil IPM would be achieved through fumigation. Some of the main results from that study were that biosolarization led to a net decrease in global warming potential by virtue of decreasing the emission of carbon dioxide. Likewise, it had a lower energy footprint by significantly lowering the primary energy demand. So we see about a 20% decrease in these metrics that speak to environmental sustainability. So why is that? If we look at the footprints for global warming potential and the contributions of different elements for each scenario, we see that in the business as usual case, the production of the fumigants is a major contributor to the overall global warming potential. There's a lot of emissions that go along with mining the raw materials for the fumigants, processing them, packaging them, and getting them to California. Now, in biosolarization, we totally negate that and instead are using more locally sourced residues from agriculture, where most of the embedded resources are assigned to the fraction of the crop that goes to use in food. And so by utilizing the residues, we're only really assigning a small portion of the invested resources to the IPM application, and we see that reflected in the decrease in global warming potential. So with that, I want to acknowledge that this has been a very multidisciplinary team. We've had investigators from UC Davis, spanning several departments, as well as cooperative extension. We've had international collaborators at Ireland and University College Dublin for the LCA piece, and we've really benefited from working alongside industry as well. Can't say enough to thank the Nicholas Nut Company for allowing us to work with them to do this trial on production acreage. That was a bold move, really driven by the sustainability vision of Rory Crowley and George Nicholas with the company. And really want to call out and thank the various researchers, students, and postdocs that have contributed to this study. Likewise, want to thank our sponsors for the work, the Western Center for Agricultural Health and Safety, which is sponsored primarily through the National Institute for Occupational Safety and Health, as well as robust and long-term support from the Almond Board of California. And with that, I also want to thank you for your time and attention today, and very happy to talk through any questions you might have.
Well, thanks, Chris. That was really terrific. For everyone on the line, you can either take your mic off of mute, or you can also type your question in the chat. I have my chat box open, so I'm monitoring that. So if there are any questions, we can certainly address those here now. If there are none from the audience currently, I will ask my question. I'm not seeing anybody with their hand up or anybody's mic off mute, so I'll go ahead and ask mine. I'm curious about this question of structural versus non-structural carbohydrates in the plant. You measured plant growth in terms of diameter, which I think is, I think pretty standard fare. But I think that's addressing, and maybe this is not correct, and that's why I was going to ask, the structural carbohydrate accumulation in the plant. And as we know, there's also this non-structural component, which primarily goes into forming yield, or is oftentimes associated with yield. And there seemed to be some evidence, at least in terms of foliage greenness, that may be occurring. And I was wondering if there was any data that you had that might be used to address that question.
Yeah, it's a good point. We don't have any data on hand now that speaks directly to that. But it's a whole nother field of metrics that we do want to capture, specifically within the fruit. There are other measures of fruit quality that would overlap with what you're speaking about, looking at the level of water-soluble sugars within the fruit, distinguishing that from the lignocellulose fraction, as well as looking at oil content, the composition of fatty acids within that oil. So there's a number of different chemical metrics that we could look at, and we intend to look at once we get more into the fruit-centric phase of the project.
Yeah, makes sense. So we do have 2 questions in the chat. I will go ahead and read those. Unless you want to read them, Chris. I guess I'll.
Yeah, I can see them here. Yeah, I see a question here. Did you make use of the anti-condensation plastic coating to increase solarization effect? No, not in this study. It's not something that is really inherent to the fumigant film, because the solar heating part is not really factored into traditional fumigation. But you're absolutely right that condensation of moisture on the inner surface of the plastic does dramatically change its optical properties, and really can increase the reflectivity of visible light, which would decrease the solar heating effect by mitigating light absorbance by the soil. So there is an opportunity, I think, to really look at different plastics and specifically the optical properties to optimize their potential use in biosolarization. The use of different plastics is really interesting to us, specifically if we can move away from the VIF and TIF used in fumigation to something that would be more renewable, like a bioplastic. But then there are a lot of questions about, could a bioplastic that's compostable stand up to all the degradative activity that we're deliberately inducing in the soil during biosolarization, in addition to really intense photo bleaching? So that's a new area of study for us, but we are trying to be very cognizant of how we can improve both the optical properties of the tarp and the sustainable sourcing of the tarp. See a question here. How did the nematode population density and damage vary by depth over the life of the study? We don't have a lot of resolution on depth. These days, we're taking 12-inch soil cores and we are aggregating the material of that core. So the data as presented really represent an average over the upper 12 inches. We do want to start taking cores from deeper down to capture the entirety of the root zone and get a profile that would be indicative of what the roots are exposed to. But right now, the data really speak to an average level over the first foot of the soil. Did you monitor temperatures? You attribute pest mortality to the organic gases, but not temperature. That is surprising to me. We did monitor temperatures at two depths, at 0 and 6 inches depth. So I didn't present those data today, but we do have them and they're represented within the article cited in the presentation. Basically, between the biosolarization and solarized treatments, we didn't see dramatic differences in solar heating. We did achieve topsoil temperatures in the 60+ degree range. So we know that there was a very potent solar heating and thermal inactivation element. We know from this study and others that the temperature does drop off pretty dramatically as you get down to 7+ inches in the soil, to a point where it's no longer probably the primary mechanism of pest inactivation, but rather is simply supporting robust fermentation and relying more on the non-thermal effects in the deeper soils, where you're probably achieving, low oxygen and more acid fermentation, which are known biopesticidal mechanisms. So yeah, for the sake of time, I didn't include the temperature data because there were not significant differences between the solar heated treatments, but those data can be found in the publication within the presentation. I'm concerned about your single row treatments. Any problems caused by this decision, such as root growth later in the study? We have not specifically looked at root growth. We had a randomized block design essentially in terms of the distribution of the rows to account for any differences in soil properties across the 10-acre site. And while certainly there has been variability, and we see that in all of our field work, it has not been enough to drown out the signals that would indicate a benefit of biosolarization. But good point on the root growth as well. There's always been a question of, was that initial 1 year apparent inhibitory period truly inhibitory, or were the trees maybe just investing more in a below ground biomass development and not so much in above ground biomass, which was really the only metrics that we were tracking at the time.
So we do have one additional question as well. Will there be an overall economic analysis of the various treatment alternatives?
Yeah, I mean, the study really begs a techno-economic analysis. And so we are in the process of doing that. It's informed by the optimal biosolarization conditions that we identify in this work and in others. So while we don't have a specific dollars per acre figure today, we do know some of the cost drivers that are involved. Specifically, one of the largest determinants of total cost is whether or not there's an existing drip irrigation system at the site. So if you can capitalize on an existing irrigation system, that is definitely advisable. If you have to install a drip irrigation system for the sake of biosolarizing, for sure it's not going to be cost effective. Other drivers of cost would be the current market value for whatever amendment you use. And we've been very deliberate to focus on residues from other food products as opposed to traditional anaerobic soil disinfestation, which is really looked at mustard meal and rice bran, which are very high value materials and in some cases are used in human food products. So looking to byproducts from other parallel industries will help to keep the cost down. And then so too with selection of the tart material and the concentration of material applied during the amendment phase, as well as the total surface area that's treated. So here we only treated the rows. There could be a total broadcast approach. Basically you can biosolarize in any way that you might fumigate. So you could spot treat as you do with fumigation, you could treat rows, or you could do a complete broadcast treatment.
Okay, I am not seeing any additional questions yet or in the chat. again, if anybody would like to just take their mic off mute, you can ask your question that way, or you can pose your question in the chat. This one is going to show my lack of knowledge about pest management, but I don't know, maybe this data exists. Do you know how the bioacids kill the nematodes?
It's not completely understood exactly the mechanisms involved with that. It's known that the conjugate acid forms are more biopesticidal than the conjugate base forms. So presumably there's some cell membrane permeability component to that. If the organic acids are abundant enough, you can have dramatic changes in pH that would probably explain the inhibitory effect. But in the soils here, they didn't really drop too far below neutral. So we wouldn't think the pH alone would be the culprit. But yeah, the exact biochemical mechanism is not completely understood.
Okay. All right. Well, then I don't feel so bad about asking that question then. All righty. I am not seeing any additional questions for you. So let's thank Chris for a terrific seminar. We will be posting the recording of the webinar on YouTube relatively quickly. Steve is out on vacation this week, and he is the one who usually does that. So it may be a little bit before we get that posted, and I certainly apologize about that. And I just want to thank Chris again for the webinar today, and let everybody know that the September webinar, IPM Hour, is going to be all about turning integrated pest management into a board game. So if you want to learn about IPM board game, the IPM board game, tune in September. Let's see the date on that. Well, it should be the second Wednesday. So that is going to be, I have it as the 14th of September. So thank you everyone for joining, and hopefully we will see you on the 14th. Thanks again, Chris.
Thank you for having me.